Now, fourth subunit of this chapter is double fertilization. This term double fertilization is coined by Navaschan. Okay. Now, so when pollen tube enters inside the embryo sac, what happens? This entry is guided by filiform apparatus, and one of the synergids will degenerate, and it will serve as platform for incoming pollen discharge. Now, one of the male gamete will fuse with egg to form diploid zygote, and this is known as syngamy. Okay. Now, another male gamete. Okay. Another male gamete fuses with secondary nucleus, which is product of two polar nuclei. So, secondary nucleus plus male gamete. So, here three nucleus are involved, that's why it is referred as triple fusion. And this triple fusion and syngamy together are referred as double fertilization, which is characteristic feature of angiosperm. Now, you can observe these are degenerating synergids. This is zygote, which is formed as a result of fusion between male gamete and egg. Then, now the central cell is referred as primary endosperm cell. Okay, and the fusion product of secondary nucleus and male gamete is referred as pen or primary endosperm nucleus, and along with that, this antipodal cell will also degenerate. Okay, and this whole event is referred as double fertilization. Okay, now after double fertilization, what will happen? This zygote will develop into embryo, and this will develop into endosperm. The embryo is 2n and endosperm is 3n. Okay. Now, the function of this endosperm is to provide nutrition to developing embryo. Okay. It provides nutrition to developing embryo. So, what happens? This embryo development takes place after formation of some amount of endosperm. Why? In order to assure nutrition to developing embryo. So, endosperm provides nutrition to embryo sorry and development of endosperm precedes development of embryo in order to assure nutrition to developing embryo. Now, our next topic is endosperm. Now, so endosperm is structure which provides nutrition to developing embryo. Now, this endosperm okay, is divided into different types on the basis of how development of endosperm takes place. Now, so it is not there in NCERT, but it is important for you people to know that. One is nuclear endosperm, second is cellular endosperm, third is halobial, and fourth is ruminate endosperm. Now, in nuclear endosperm, what happens? This primary endosperm nucleus undergoes successive nuclear division, give rise to free nuclei. That means here karyokinesis is not followed by cytokinesis. So, there is development of free nuclear endosperm. Okay, and after that there is formation of cross wall. In case of coconut, okay, coconut water is nothing but it is free nuclear endosperm. N number of time question have been asked from this part. In case of coconut water is what? So it is nothing but it is free nuclear endosperm. The coconut water from the tender coconut that you are familiar with is nothing but free nuclear endosperm. Okay. Now, then second is cellular. In case of cellular, what happens? Every nuclear division or every karyokinesis is followed by cytokinesis. Okay. This type of development is observed in Solanaceae. Now, In case of halovial endosperm, which is observed in case of monocot, what happens? Part of endosperm is nuclear and part of endosperm is cellular. Part of endosperm is nuclear and part is cellular. Now, ruminate endosperm is observed in case of plants such as Myristica, commonly known as nutmeg, Annona, Serifa. Okay, here what happens? There is unevenness on the surface of endosperm, and this unevenness is due to either activity of endosperm itself or due to the activity of seed coat. So either activity of endosperm itself or activity of seed coat will cause unevenness on the surface of endosperm, and such type of endosperm is referred as ruminate endosperm. Now, after the development of endosperm, what will happen? Development of embryo will take place. Okay. Now, what happens? This is zygote. Now, this zygote first divides by transverse division into two cells. Okay, this is apical cell and this is basal cell. First, this basal cell will divide, okay, to form a structure referred as suspensor. Now, purpose of suspensor is to push the developing embryo inside the nutritional environment of endosperm. Then further development and differentiation will take place, and that results in formation of heart-shaped embryo. Okay. Then finally there is formation of mature embryo, which consists of plumule, radicle and cotyledon. Now, there are 4 different stages in development of embryo, that is proembryo, globular stage, okay, then heart-shaped stage, and finally mature embryo. Okay, so development of embryo up to proembryo stage, or we also call it 8-celled stage, is similar in case of both dicot and monocot. Okay, up to 8-celled stage, it is similar in case of dicot and monocot. Okay. Now, these are the 2 diagrams of dicot and monocot embryo. First, it is dicot embryo. Okay, this is your dicot embryo, which consists of 2 cotyledons. Okay, these are 2 cotyledons which are heavy due to storage of food material. This is plumule, this is radicle, and this is root cap. Okay, now part of embryo above cotyledon is referred as epicotyle, and below cotyledon is referred as hypocotyle. This diagram is very important. Question have been asked from this diagram. Suppose someone can remove this labeling and I will ask you what it represents. This blank, fill in the blank. That is nothing but radicle. Okay. Now, next is monocot embryo. This is your monocot embryo. So a monocot embryo consists of single cotyledon, which is large and shield shaped, and that is referred as scutellum. So in case of monocot, cotyledon is referred as scutellum. Then we have shoot apex, that is plumule, and radicle, and both of them are protected by undifferentiated seed, that is coleoptile and coleorhiza respectively. Then in case of grasses, there is a structure, this question have been asked, which is a remain of second cotyledon. Remain of second cotyledon, and that is referred as epiblast. So this is structure of monocot embryo. Okay, now I will discuss previous year question from this unit, that is unit 4. Now, first question is, what is the fate of male gamete discharged in the synergid? Okay, so two male gametes are discharged. One fuses with the egg, true, and other fuses with central cell nuclei. Okay, central cell nuclei. That is two polar nuclei which is present, which is referred as triple fusion. So this is true. Second choice is one fuses with the egg, other degenerate. Not true. Third, all fuses with the egg. Not true. One fuses with the egg, another fuses with synergid nucleus. Not true. So correct answer will be first. So answer is first. Next question. Coconut water from a tender coconut is nothing but it is free nuclear endosperm. Okay, it is nothing but free nuclear endosperm. Answer will be second. This is very important question. Large number of times this question have been asked. Now, again, the coconut water and the edible part of coconut are equivalent to, it is equivalent to endosperm. First choice. Okay. Next question. Double fertilization exhibited by, double fertilization is characteristic feature of angiosperm. Okay, so answer will be third. Next, double fertilization is fusion of one male gamete with egg nucleus, another with two polar nuclei. That is fusion of two male gamete of a pollen tube with two different egg. No, fusion of one male gamete with two polar nuclei. Not true. Fusion of two male gamete with one egg, syngamy and triple fusion. So correct answer will be fourth. That is syngamy, that is formation of zygote, and triple fusion, formation of primary endosperm nucleus. So answer will be fourth. Next question. Now, what would be the number of chromosomes in cells of the aileron layer in plant species with the 8 chromosome in synergid? So synergid is haploid. So n is equal to 8. And aileron layer is part of endosperm. So aileron layer is 3n. So value of n is 8. So number of chromosomes will be 3 into 8. That is 24. So answer will be second, that is 24.